All right, everybody. Good morning. It is May the 4th. Today is the initial day that the quarantine was going to end. Imagine going back to school right now. Yeah, well, that's not the case. All right, so today's lecture is going to be on DNA transcription. Right. And so just a quick recap. Last week, we went over DNA replication, which was making more DNA based off of your original DNA strands. We learned about adenine, thymine, cytosine, and guanine, your nucleotide base pairs, and which ones go together. All right. A's and T's go, and then C's and G's go. Sorry. So that's pretty much last week in a nutshell, all right? And so we're going to be using that and moving forward with it. So I'm going to present my screen, and we're going to go over DNA transcription, right? And so in short, all right, transcription is step 2, all right, of this process, all right? Typically, when you go from DNA to proteins, the 3 steps are replication, we did that last week, transcription, which we're doing now, and then translation is what we're going to be doing next week, all right? So just going to go through this PowerPoint. All right, so central dogma, all right, that just means like, the main rule, all right, and so up here it says DNA to RNA to proteins, all right, that's exactly what I was just saying. So DNA to RNA, well, that's what we're doing today, it's called transcription, and then RNA to proteins is called translation, all right, and then Chargaff, we learned about him, he discovered that the 4 nucleotide base pairs are pretty much present in DNA in equal amounts, all right. So we do have a couple different types of RNA, and I'm going to make an analogy right now, okay? So the nucleus was the control center of an individual eukaryotic cell, all right? Go a little bit bigger and think about what controls us as the human body or human species, all right? The brain and the central nervous system pretty much control what we do day to day. And the way they control us is by using RNA signals going throughout the body to essentially dictate what it wants the body to do. All right. And all of this information in RNA is derived or comes from DNA. Right. I'm really trying to work these bigger concepts down into one video. So sorry if it seems like I'm not explaining too much, but I just don't want to throw too much miscellaneous information in here. But there are 3 types of RNA, all right? Messenger RNA, all right? Ribosomal RNA and transfer RNA, right? Each has its own function. You can read those there. But the main ones that you're going to hear are the mRNA and the tRNA, right? So Griffith's experiment, all right? In short, all right, what he did is that he found out that genetic material doesn't exactly go away. All right, we found out when we were going through biochemistry that when you heated up a protein solution, those proteins denatured, and specifically the enzymes that we were working with in that experiment could not work anymore. And so what he did is that he found a strain of virus. One was known to kill the host and one was not going to kill the host. All right. And so on the blue and red left 2 columns, those are just normal injections of just the virus. All right. The blue treated mouse lived, non-virulent, and the red treated mouse unfortunately died. All right. And so he tried to do the same thing. He heated up these viruses and saw if they could still work. And so he heated the smooth bad strain, all right, and it didn't kill the mouse, all right. And then what he did is that he mixed the rough strain, not heated, and the heated smooth strain. And so the genetic material from the heated smooth strain was still there, and it was taken on by the rough strain, the normally non-violent or non-harmful strain. And essentially, it just took on the genetic material from the more harmful virus and took it on. And so this experiment, and the proof of this experiment was in the rightmost column, is that that mouse died, right? He proved that genetic material can be passed not only from region to region within solution, but that genetic material kind of never goes away, right? Avery, he actually did more research on proving what was our genetic material. He pretty much proved that DNA was DNA, essentially in a chemical way, right? That's pretty much on Avery. He was the biggest one, but Hershey and Chase, right? Again, they manipulated some bacteriophages and such, and essentially they just proved and pretty much captured what they look like. All right. Chargaff, again, we learned about the nucleotides in their proper proportions. All right. And just because it's in front of us right now, I am going to bring this up. All right. So when we're dealing with DNA, our four nitrogenous bases were adenine, thymine, cytosine, and guanine. A, T, C, G. That was DNA. Now we're moving into RNA. And there is one difference. And that difference is, instead of thymine, right, when you're dealing with RNA, thymine is replaced with a nucleotide called uracil, right? And so a little bit later, I'm going to go through an example, but essentially wherever you would put a thymine, you are now going to put a uracil when you are making an RNA strand based off of a DNA original, right? Purines and pyrimidines, all right? Those were vocab words. All right, transcription and genetic code. All right, so there is essentially these numerous amounts of chromosomes in each given cell, right? But not the entire length of the chromosome is going to need to be copied into an RNA complement every time. And so when the body or the cell wants to create a certain type of protein, right? Because that's the end goal, is to make a protein. On your DNA strand that's going to be copied, there is a promoter region, which essentially promotes that region to be copied, right? You can probably make that connect the dots, right? And so in the process of transcription, what we're going to find out is their name is DNA polymerase. It's an enzyme. It's going to find that promoter region and bind to the DNA. And we learned that DNA was a double helix, all right? And that means it's a ladder structure that's twisted, right? And so when DNA polymerase binds to that twisted ladder, it actually untwists a little bit, and it actually pulls the ladder apart, right? So it splits those ladder rungs right down the middle, all right? And it essentially holds those two pieces or two sides of DNA open, so it can start to make a strand of RNA based off of the complements to one side of that DNA, right? We know genetic code, codon, right? Codons come more into play with translation of, building amino acid chains. But just because it's here, it's just a group of 3 nitrogenous bases. Right, so if your DNA strand goes ATG, that is a codon. Another codon could be CCG or CCT. All right, codons come more into play again in translation. All right, steps. All right, so the initiation step is when that DNA polymerase is going to look for that promoter region. All right, and once that DNA is bound to by that RNA polymerase, we are going to read those base pairs and essentially just make an RNA strand complement, right? Elongation is just making the strand, all right, and termination is when you started at the promoter region, and then you have made your RNA strand by reading consecutive base pairs. And then once you have a terminating region, that RNA polymerase is going to unbind, and now you have this copied mRNA strand. But you're not done yet, all right? We do have this piece called introns and exons, all right? And there's not part of this PowerPoint, so I'm going to stop sharing and just briefly, all right, explain that. All right, so again, we have coding and non-coding regions of our DNA, and the coding regions are called exons, and the non-coding regions are called introns. And those are just everywhere in your DNA, all right? And essentially, the step I'm going to explain is just kind of a cleanup step. And long story short, you just made an RNA strand based off of just a raw DNA strand complement. And that DNA strand had important and non-important coding and non-coding regions of that strand. And so now you have this mRNA strand that is eventually going to be made into a protein, but pieces of that strand you don't want there. And those are the pieces that were made from a complement of an intron, that not coding region, right? And in a very, very simple way, you get some enzyme activity, and they essentially are just going to cut out the introns, the non-coding regions of that RNA complement, and they're just going to cut those out and then stick the exon complement regions together, and now your RNA strand is strictly going to copy for the protein that you want. All right. So I am going to stop the video here, right? Again, I encourage you to ask any questions that you had come up, right? They can be about the silly haircut, or they can be about transcription, all right? But I do encourage you to kind of broaden your mind on this. So I'm going to be posting accompanied YouTube videos that I would love for you guys to watch, right? The assignment that I'm going to be posting is just a single page Word document. It is not very difficult, all right? So this is the lecture video for now, and I hope everyone has enjoyed the nice weather that we've been having lately. And yeah, keep with it, guys. All right, peace.